Good evening and welcome to the York Festival of Ideas. And it's a sort of evening edition, and we're all delighted that you're here with us this evening. My name is David Kent, and I'm a professor of stem cell biology at the University of York at the Center for Blood Research. And tonight we're really delighted to have with us a wonderful speaker. You don't want to listen to me all night. You've obviously tuned in to welcome our speaker, and that is Dr. Coleen T Murphy. And we're really delighted to have you here with us this evening, Coleen. Coleen is a professor of genomics and molecular biology at Princeton University in the United States. She's the director of Princeton's Glenn Foundation for Research on Aging and the director of the Simons Collaboration on Plasticity in the Aging Brain, and also director of the Lewis Sigler Genomics Institute at Princeton University. So she spends all of her time directing. But today she's going to direct us through her latest book, and that's on how we age, the science on longevity. And Coleen will look forward to your presentation, but also to the interactive question and answer period following that. And so without further ado, really, Coleen, welcome, and over to you.
Thank you very much, David. And thank you for the very kind invitation. I appreciate the organizers picking my book to highlight and to get to discuss this with all of you. Okay, so I want to tell you tonight about how I got interested in the topic of aging and what we know about it. And really, this book is not about what to do about aging. So it's not really a self-help book. What it is giving you an idea as the reader of how we even came to the ideas that we currently hold in the aging field. So, you know, all of us probably would like to live a long time, right? So the most famous example of this you may know is Jean Calment. She was allegedly lived to be 122, although some interesting stories about why that may or may not be true. Even if she was not the longest living human being known, we do know that others have lived to be almost that long. So Sarah Knoss in the United States and Kana Tanaka in Japan lived to be almost 120 as well. And so that's kind of the upper limit of what we know of human lifespan presently. Now, what you should keep in mind is that is in the absence of all the research that we're gathering now in ways that we might be able to extend lifespan later. But of course, all of us have in the back of our minds also this idea is longer, always better, right? So really, what most of the field is focused on right now is not extending lifespan to the maximum, but really extending health span. So some of what I'm going to tell you tonight deals with that idea, keeping us healthy the longest time possible. Because we really think that quality of life, not the exact quantity, is probably the most important thing. We always think about that because, of course, there's a downside to a longer life if you're unhealthy. And so we really want to keep track of ways to make a healthy life extend longest. Okay, so in the book, I talk a little bit about the ethics of longevity because I've had a lot of people ask me that, especially when I first started working on aging. But I think it's very simple to say it in this way. If we figured out every way to cure cancer, cure heart disease, cure every single other age-related disorder, we probably wouldn't have a problem with that. And so really the only problem that people have is when we actually just talk about extending lifespan. So I think that ethics is not really an issue here. We just have to think about what are the wisest ways to plan for a long lifespan that is equitable among everyone. So what I'm really going to focus on tonight, and really the bulk of my book talks about what do we know about how longevity is regulated, really at the molecular level. And then at the end of the book and at the end of this talk, I want to tell you about what we know about how far we are away from real treatments for aging and age-related diseases as a result of the research that's been done on aging. Okay, so just to back up everything, it's a really great thing that we are living much longer, right? So on average, lifespan is getting longer, and in fact, by year 2050 in the U.S., at least, a much greater percentage of the population will be over the age of 65. And of course, the problem with this is that a lot of age-related disorders come along with increased age. And so the general idea of the lifespan or aging field is that if we could slow aging, we might be able to have an impact on a lot of these other age-related disorders that decrease our quality of life. All right, so what's considered old? All right, it really depends on your perspective. Now, for a US audience, this is usually a more the idea of what's an old person. For example, who is most functional? It kind of depends on what they're doing, right? So in the US, a football player named Jerry Rice was quite famous for a while because he was very old for a football player. He was in his 40s when he finally retired. Andre Agassi as well. Gymnasts are generally, and actually until quite recently, they were kind of washed up by the time they turned 20, and that's only recently changed. But certainly when I was a kid and when I was a gymnast, hitting 20 was even considered old. So it really depends on that function. Of course, for scientists, we can do this almost forever. So it's not really a problem for my field. So what is considered old depends on function. And that kind of resonates all through our research on aging because it really depends on the function of, for example, every organism that we look at, what is the function that we're looking at to decide what is considered aged. All right, so a little background about me. I'm actually not one of these people who's been obsessed with the idea of aging ever since I was a kid, which is what I read from incoming students all the time. Actually, I was a graduate student at Stanford University where I worked with Jim Spudich. And the question that we were really interested in was, if you have a crystal structure, so you have a protein structure of a motor protein, can you understand how a motor protein, like that molecule, turns energy into movement? And so I was really obsessed with this, working on this one protein and its interaction with another protein. I got very good at asking questions at that molecular level. By the time I was looking for a postdoc, I really wanted to expand the kind of questions I was asking. And that was right about the same time that Cynthia Kenyon at UCSF had these remarkable papers about aging and lifespan, where she used this small organism called Cinereptitis elegans, which is shown here in the bottom. This is really just, this animal is only 1 millimeter long. And I'll talk a little bit about what Cynthia found. She made discoveries that found that she could find animals that lived twice as long. These needles live twice as long as their normal counterparts. And so that really excited me. I thought, well, this would be a great question to go and ask about aging, because it's kind of the opposite of this like single, like little tiny molecular question between two proteins. And that, you know, frankly, if I went to a party, nobody ever cared what I worked on. But as soon as I started talking about aging, people were like, oh, that's something I want to know about too. Okay, so before I went to our lab, I started thinking about, well, what is really the good way to study aging? Okay, now I just showed you some pictures of centenarians, and you can take the genomic information from centenarians. We can do this now. We've gone so much better at looking at the genetic code and the sequences in all of our DNA that we can actually do what's called genome-wide association studies. And that means that if we collect these centenarians of very, very long-lived individuals, we can look at their genomes and find things that are different from them to people who have a relatively normal lifespan. And so that's one perspective you can take, and a lot of great researchers are doing that. We can also look at extremely long-lived animals. So that's another way to go. For example, the bowhead whale lives to be over 200 years old. So that's the longest lived mammal. And so by looking at its genome and the proteins that it makes and how its cells function, we can get some clues into how, for example, if you compare it to a relatively close relative, another species of whale, we can pick out things that are different between those and ask, well, maybe those are the reason this animal is able to live so long. Of course, we want to test those theories. And so to do that, usually we go into the lab and do things in a tissue culture dish. So we'd look at cells at sort of that cellular level, not inside the animal, but it allows us to test our theories at the cellular level. And so that gives us a lot of information as well. What I was interested in doing, the reason I went to Cynthia Kenyon's lab and it's really guided the work that my own lab does since, is instead sort of combine all these, where you can do this, molecular level kind of questions, but in a whole animal, so that you can understand what is happening in this animal that allows it to live long and function better, but can we actually get at its genetic reasons as well? Pardon me. Okay, so give you some examples of some kind of cool stories about longest lived animals. A lot of these longest lived animals on our Earth, besides trees, so trees are actually the longest lived organisms on Earth. But if we go to animals, we see that the sea is really where they live. And so, for example, we have the Ming, the Icelandic clam. So this clam was caught, and when it was cracked open, which unfortunately killed it, it was found that that clam was found to be 507 years old. Okay, so that's remarkable. That was done through carbon dating studies. So it's clear that these these clams can live a very long time. And when later studies were done on the same species, found that there's no significant oxidative damage to its heart, which is interesting because it means that these animals are somehow like turning on mechanisms to prevent that kind of damage as they live a long time. And here on the right, I'm showing you the Greenland shark. So this is not a mammal, it's a vertebrate though, and it lives super long. It's swimming in very cold, dark water. So the idea might be that it might have ways to slow its metabolism and thus slow damage. All right, so the reason I love this is because I think it's kind of cool. The way they date these is they take eye tissue because the lens of the eye doesn't ever turn over. And so you can actually carbon date that to figure out how old the animal is. So they're examples of these super long-lived vertebrates, right? And then we have animals that live on land, terrestrial animals. So there's Jonathan the tortoise. This is probably actually a little, it might be even 192 years old now. So this animal and some of its relatives live, they're from Seychelles, and they live to almost be, now it'll be 200 years very soon. Okay, so we don't know exactly why, but they're living very long. All right, so I mentioned the bowhead whale. That's the one that we actually know from actually looking at things like having hunting gear removed from it. So we have to understand, like, what is the reason that these animals will live a long time? Okay, so let me just go back up. So one problem with working these super long-lived animals is they're not experimental organisms. We would not want to start doing experiments on bowhead whales, right? So we have to take all our information from observations, and in some cases by collecting tissue and doing things like genomic analyses. The other way to think about aging is not to think about like in the scale of years, but to ask relative age. Like if you're a small animal, maybe you aren't capable of living 200 years, but maybe you could live much longer than another animal of a similar size. So this is called a longevity quotient. And the idea here is that as animals grow larger and larger, their lifespan roughly changes as well, right? So elephants are fairly long-lived, mice are fairly short-lived, but there are exceptions to that. And that's what you hear about in the news often. For example, we hear about the naked mole rat. So that's an animal that can live something like 30, 35 years, even though it's about the size of a mouse or a rat. And so we talk about that because that's a really exceptionally long, high longevity quotient. Similarly, bats, some species have little tiny bodies, just like a mouse, but they can live 30 years as well. So we can do these comparative studies between animals of these different longevities and to try to figure out what might be different. And so what I'm telling you is there's a whole bunch of different ways that scientists use to study longevity, all the way from, humans, I told you about GWAS studies, but all the way down to super short-lived animals. And the reason I'm going to tell you about this is because even though it's really exciting to study an animal like the naked mole rat that lives for 30 years, it's really hard to do an experiment, right? So we have graduate students in our lab for 4 or 5 years. They need to actually be able to do some work. And so to carry out experiments on, for example, naked mole rat or primates, for example, that also live for 30 years, it can be really difficult. And people do that, but generally, we try to look over here at the other end of the scale to ask, can we do really fast experiments, okay? So what I'm showing you here is this scale of years, time rather, and at one end we see there's bacteria. So bacteria live for a couple hours, and they're single-celled organisms. So we can learn something from them, but they're prokaryotes. They're pretty different from us. They're more like our mitochondria. And so there are experiments on aging that have been done in yeast cells. So those are true eukaryotes. But they're also often single-celled. And so we have to also limit what we learn from them. But the vast majority of work that's being done in the aging field has really started with these very simple model systems, C. elegans and Drosophila melanogaster. And the reason for that is that they only have very short lifespans on the order of several weeks, but we can do a lot of experiments on them really quickly and we see them age. So we actually know that they're aging and we can ask questions about the process of aging. And what's really important here when we're talking about C. elegans is the fact that a huge fraction of the proteins that are made by these animals are exactly like the proteins in us. All right, so if that had been not true, for example, if C. elegans had been so wildly different from humans, then it may have been of limited importance how we, what we learn about aging in these animals. But for the most part, when we find something that regulates an important process related to aging in worms, we often find that there's a homologue in humans as well that may have a similar function. So I think that's really important to understand because, often when you hear something in the news, you'll hear about naked mole rats or work that's been done in mice, but often the base work has been done in simple systems like C. elegans and Drosophila before people have tested them in longer-lived, harder to work with animals like mice. Oh, I do want to draw your attention to one really cool animal. There's a turquoise killifish, which is a fish that was found. It's in African areas where there's rainy and very dry seasons. And this is the shortest lived vertebrate. So I don't work on killifish, but I'm very excited about the work that's being done in this area because it really is going to be able to open up a lot of new areas of study because it's a vertebrate. And so it helps us understand what might be going on all the way, in a way that maybe we won't be able to do in Drosophila and C. elegans. Okay, so now we're going to get more to these are the tools of the field, more up to a philosophical question about how we study aging, okay? Now for years, people asked the question, what happens with age? And they would compare young things with old things, young animals with old animals, and kind of basically, document, catalog the differences, right? But the problem with that, of course, is that you can't really know what's causing aging, right? So if I told you cataloged all the differences in these photographs, one of them where you'd say well, this person has dark hair and this person has gray hair. And is that a cause or an effect? Well, we all know that gray hair doesn't cause aging, it's an effect of aging. But if you were talking about two proteins, you wouldn't know the difference, right? And so it's really important in the field that we do make these distinctions between causes and effects. So the only way, or the best way to do that, is to ask the question about mutants, all right? So can we take two animals that are similar age and one functions younger than the other one, or can we take animals and make one tiny change in them, and does it change their lifespan? All right, so this is really a tool, and the reason I went to Cynthia Kenyon's lab was because she was really the first one to show this effectively, that you can make these small changes in the genome and actually get something very different with respect to age. Okay, so let me tell you what I'm talking about. All right, so I'm going to mention C. elegans a lot in this talk, but it's because what I work on, it's really what's brought a lot of information to the field. So these are tiny, they're microscopic, they're 1 millimeter long. They grow up in about two to 3 days from an egg to an adult, and they have a very short lifespan. It's only 2 to 3 weeks. But any of you who are, if I gave you pictures of a young worm and an old worm, you'd be able to tell the difference, right? They have really obvious aging phenotypes. They slow down, they get kind of fat and dumpy and eventually paralyzed. So they really are, it's really obvious that they're aging. So this is a great tool for us, right? Okay, so this is a movie of these animals. And so this is a normal worm. It's a young adult, what we call wild type. All right, so it's crawling around on its food. It's swimming around on a plate of bacteria, which is its food. And there's a mutant that I'm going to tell you about. This is what Cynthia Kenyon discovered. It's called DAF2, and I'll tell you about that in a minute. And on the first day of adulthood, you would not be able to tell the difference between DAF2 mutants and wild type. They look exactly the same. But, about 13 days into adulthood, a lot of wild type animals are looking very sad, right? So this guy's still alive, and we use a pick. We would poke it in the head to make sure it's alive. Actually, it's still kind of moving. Sorry, this part of the movie is a little slow because we have to always make sure that everybody understands that these animals are actually still alive. So, well, at least the one on the right here is getting very paralyzed. The one on the left, we'd really have to poke it. Okay, that DAF2 mutant on that same day of adulthood looks like this. Okay, so again, anyone in the world who just looked under the microscope at these animals would be able to tell you that this thing lives, it looks young, it's great, it's happy and vigorous. And so that is what really, to me, changed the whole field of aging. Oops, I'm sorry, let me make sure I can go. Okay, so this was a mutant that Cynthia Kenyon discovered. It's called DAF2, and this is what's called a Kaplan-Meier survival curve. And you can see that these DAF2 animals live twice as long, and you saw the movie, they're very healthy, right? Well, it turns out when Gary Wilkins' lab cloned this animal, the mutation, it affects the insulin receptor. Okay, so as soon as you hear insulin, you're not thinking like this is a weird worm thing. This is actually like something that could affect us as well. And it turns out it does. Because it turns out some of those genome-wide association studies of centenarians identified genes in that same pathway, including the IGF-1 receptor, as being correlated with exceptional human longevity. So again, what we find in these mutant or these model systems they're really relevant for us as well. And the way this works genetically is we would say, okay, a mutant blocks this path. I'm going to teach you some small, like a little bit of genetics here. So the DAF2 mutant, its normal function is to block longevity. So when we turn it off, now you get longevity. And it requires the activity of another gene called DAF16. So normally DAF2 turns off DAF16. DAF16's job is to turn on longevity. Okay, and so you can see when you take away DAF16, they have a short lifespan. And that's because DAF16 is what's called a FOXO transcription factor. It goes into the cell and into the nucleus, and it sticks to genomic DNA, and it turns on the expression of a lot of genes that help us out. Okay, so the way this works, so we have DAF16 turn on all these genes that enhance our lifespan or the worm's lifespan, things that fight stress, antimicrobials, lipid and steroid metabolism. It turns out, just like in us, fat metabolism is super important for regulation of health. And it turns off genes that actually would be bad for longevity. And what's really interesting is the cell always has to decide between living a long time and growing and developing. And so that is a little bit of a clue for us that cells can't always do it all. They really need to make these important choices. And sometimes stress resistance versus development is the big choice that it makes. Okay. And so, sorry, let me just highlight this. And you'll probably ask me about dietary restriction. Similarly, a lot of dietary restriction pathways, what they really do is they tip the balance in favor of stress resistance as opposed to something like growth and development. So speaking of dietary restriction, all right, so this is these are pictures from a famous study that was done a few years ago. I believe these pictures were from the Wisconsin group. And what they did was they took rhesus macaques and they fed them either a normal diet, which turned out to have been a high-fat diet. And those animals, they get old, and you can see that on the left, they're aging quite a lot. Same aged animals that were given dietary restriction, that means they had to reduce their intake about 30%, had a really improved health span, right? And they just look healthier, right? This pathway is really well studied. There has many downstream effects that look a lot like what I was just telling you about. And there's also, I think especially in England, intermittent fasting is very popular. So intermittent fasting shares a lot of the aspects of dietary restriction. And one of the exciting things, I'm not myself very excited about doing dietary restriction. I really like eating and I like enjoying life. So what I'm kind of interested in is the idea that there are what's called DR mimetics, so drugs that may mimic dietary restriction that are being developed. And it's a really active area of research. And one of the ideas in the field is that mTOR inhibition by rapamycin, it might be one of these dietary restriction mimetics. So many of you may have heard of rapamycin. It's actually very popular. We'll have to see what clinical trials turn out with for a health span, but that may be a way of mimicking dietary restriction without carrying out yourself. Okay. All right, so one of the important aspects of studying aging is trying to figure out if you're actually seeing a difference or not. All right, so the reason I'm showing you these pictures here on the left is we have many ways of judging whether a person has aged or not. Right, so when we see someone walking, we kind of have a sense of how old that person might be. When we see their faces, we have a very good idea. And so those are considered biomarkers of aging. These are like visible ways of tracking how long someone ages. I'm showing you this picture because it's very famous. Some of you may recognize this picture. This woman, she actually was found later, about 17 years later, and she had aged considerably. She'd lived a very stressful life and was outdoors a lot. And you can see really marked evidence of her stressful life and aging. So if we want to take biomarkers and really understand biomarkers with age, we'd really like to understand what are some of the biological markers of aging rather than just chronological age, right? Because that's what we want to get at. In fact, if we could find really great biomarkers, it wouldn't just correlate with the age that someone is, it would actually tell us how long someone is going to live. And a really good use of those biomarkers would be to assess whether, there's all these interventions that we might try to ask ourselves, are we slowing aging or not? And to understand this, we need to understand the difference between biomarkers that affect a specific tissue versus something that would affect the entire system. And so the idea with these biomarkers is that we might not be able to untangle cause and effect, but at least we'd be able to see which biomarkers correlate with aging and slowing age rates. And so the idea in the field is that we would be able to use things like proteins, for example, proteins from our blood or hormone levels, or even most recently, the idea of biological clocks. So maybe there's something with the DNA and methylation of DNA that we could use to track how fast we are aging. So you just saw a picture, of some evidence of aging through the face. This is actually a very active area of research. Pardon me. And in fact, Jackie Jindong Hong's lab a few years ago did a really great analysis where they developed a way of doing 3D analyses of faces. And so by looking at a huge population, so Jackie was in Shanghai at the time, so these are all Chinese faces, but she was able to look at these huge set of populations and do these 3D imaging, and then be able to track which of the many parameters that you see in a face that change with age, which ones actually correlate best with age. And so this was super exciting because you could actually then use this to test intervention as well. And in fact, in this paper, they show that these changes in the face with age are actually better predictive of aging than anything that's been found yet in blood factors yet. So I think that this is going to be exciting. I don't know how commercially this will be applied. I'm sure someone has a great idea. But it's kind of interesting to think that it matches our intuition that we see faces change, and you can actually track this very well. At a clinical level, we want to be able to assess how frail or healthy people are when they get to a later stage of life. And so gerontologists have used this short physical performance battery for several years. Actually, it's not that young anymore. It's from the 90s. And it's very straightforward. So patients will do something called a repeated chair stand. So they just stand up and down. They also balance. And then they do this 8 meter walk. So maximum gait. Okay, so this is actually really predictive of how long someone will be able to stay out of a hospital and how healthy they are and their mortality over the year after the hospital discharge. But of course, this is for very advanced ages. What we really would like to develop is something that's similarly simple that we could do that would predict a future lifespan in people who are middle-aged, for example. And so that's a kind of cool question. Could you actually have people do like a 50-meter dash or something in like when they're 45 years old and see how well that correlates with how long they live? We were kind of interested in this question with worms because we can do the experiments really quickly. And so we wondered, we can't do repeated chair stands or balance testing with worms, but we can ask how fast they might be able to sprint. And so my colleagues here in South Korea, and we collaborated to do a pretty simple, straightforward experiment. So basically we have worms crawling around on the plate, like I showed you with bacteria. Every day, Dr. Hom here, he would put every single individual worm onto a plate with no food, okay? And the reason that's important is because when they have no food, they run around fast looking for food. They really want to find it. So we video recorded them, each one of these individual worms for 30 seconds every single day and put them back on the plate. And then looked at their lifespans. And what we saw is if you track each one of these individuals, of course that changes with age, but the thing that's most predictive is the maximum velocity. For example, if the worms are crawling around and they sprint for a little bit, that maximum velocity is super predictive of their future lifespan. And we know that because what we did was we took worms later, we tracked their maximum velocity up to day 9, and then we split them into the slow ones and the fast ones. And we can see that the worms that have a really high maximum velocity end up living longer, much longer than the worms that have low maximum velocity. And so this is really a great way to distinguish, even in worms that are genetically identical, what might their future hold, okay? I'm excited about this because I've been talking with some of my colleagues. So in Drosophila, a colleague of ours have been able to figure out a similar test that's kind of a complicated grooming behavior. And it turns out on the first day of adulthood, this is Grace Mackenzie Smith and Josh Shavitz's lab, might be able to predict how long those flies can live. And similarly, Anne Brené's lab has work in killifish where they've filmed the fish and watched their behavior. And they've been able to figure out a behavior that correlates with their future lifespan as well. So because this works in all these different organisms, it suggests that there may be something in humans that might work as well. Okay, so I've told you about a bunch of different kind of basic ideas that I cover in the book. I'm not going to go through each one of these in great detail, but I want to let you know that some of the ideas, there were some really big surprises in the aging field over the years. Some of them, and I am going to explain some of these. One of these is the sensory regulation of longevity. That was a surprise. The idea that your germline might regulate longevity as well. My lab discovered that mating can affect lifespan. That was kind of horrifying. We also discovered that reproduction and lifespan are linked. So evolutionary biologists like to think about how reproduction and basically other life history traits are linked. But we were able to show that reproduction and lifespan are linked, but they're actually regulated separately. I mentioned earlier these molecular marks like methylation groups. So there's molecular markers that can be used to develop these clocks. And so actually, these things are being sold now. If you were to go online to look for DNA methylation clock or clocks to measure, you can actually send your blood off to get measured. I don't know how well any of them work yet, but it's already being commercialized as a way to measure your true biological age. And then a few more points. There's factors in blood that can affect aging rates. I'll touch on that in a little bit. And then several of you maybe have heard that the microbiome may affect aging as well. Okay, I'm just going to do 1 or 2 slides on each one of these points. Okay, so one surprise is if you add all these pathways together. So I told you that worms live 2 to 3 weeks. That's the black line here. Cynthia's lab years ago was able to add up, basically accumulate separate genetic longevity pathways and show, and this is a movie of some worms that are 144 days old. Nuno, who worked on this, he named the last 4 after the Beatles and then became correspondingly dark after that. So anyway, they can live a long time if you add them up, all these separate pathways. Okay, so just to get back to this weird idea, can something that you sense actually affect how long you live? This seems really absurd, but the more we learn about it, the more it seems possible. And so we have to think about if they do this, then why would it work? Okay, so I'm not going to go into a lot of worm neurobiology. We've known for a long time that if you use a laser to kill certain neurons that are responsible for sensing the outside world, like smells of food, that the animals can live a long time. And that's shown here on the right. So basically, animals that can't smell their food anymore can live a long time, okay? And then animals that have also lost their sensory neurons that regulate with their germline also live a long time. So the idea here is kind of like more we think about it, the more it makes sense. That you could have an animal that's sensing its outside world and making decisions about whether it should reproduce or not. Okay, so that kind of sets the rate of aging clock for them. If they don't sense their food, like because they can't smell it anymore, then maybe it would tell the animal's body to slow down reproduction because there's no food to be eaten when it lays those eggs. And so it should slow down reproduction and therefore also slow down the rate of aging of the rest of the body. Okay, so this is how we can think about this coordination between sensing the outside world, making a decision about whether you're getting enough nutrients or you will get enough nutrients to reproduce. And then if you're not, then slow down aging. Okay, so again, this maybe seem like something that's just specifically for worms, but other researchers went on to really go into great detail and not only study this in the worms, but find similar or even homologous mechanisms in Drosophila. So same idea, if they can't smell their food, then they live longer and they slow down reproduction. And even in mice. So in mice, it was shown that a pathway that senses at least pain and functions within the CRTC1 pathway can actually slow down aging as well. So, as a field, we're still trying to figure out all the different ways that sensing the outside world may have an influence on rate of aging and especially reproduction. But I think this is not just not the only thing that we'll find. Okay, I'm just going to share with you one of my favorite papers this is from a Drosophila lab, Scott Pletcher's lab. And he had this amazing observation about stress and longevity in flies. So what he found was that when he had a bunch of flies that they basically are exposed to other flies who are dead, okay? So I'm just trying to figure out this picture over there. They have to basically hang out with a bunch of dead flies. It stresses them out and they die sooner. Okay, so and they did all this cool work. They like painted the eyes so they can see, they show the effect goes away. So it really is when the flies see another dead fly and they can't escape, they live shorter. So I think that's really cool. So, and it's not about the odor, and it kind of makes sense. If you're a fly and you see a dead fly who looks like you, and that's the other cool thing, they did it with other species that were different sizes. So the flies know who they're related to. And so if they see flies that are dead, who are definitely related to them, they're like, I need to get out. They don't care if they're flies that are flash frozen, so they don't actually look dead. So they have to look really dead. And they don't care if they're other species. They're like, well, it's probably not going to affect me. So anyway, so in that case, the sense of vision really does seem to affect lifespan in Drosophila. Okay, I'm going to switch gears a little bit and tell you about some of the kind of stories we love to tell in the longevity field because they're kind of crazy. All right, so I don't know if any of you watched this, but it's the same idea. So heterochronic parabiosis, what's that? The idea here, and it's a really old idea, it's actually developed in the 1800s, and the idea was to take two animals, and if you stitch a young animal together with an old animal, kind of remarkably, they start to share a circulatory system, all right? This is true if you stitch same animals together too, but that would be isochronic. So this is heterochronic parabiosis. So the idea is that the circulatory system then becomes shared between these two animals. And what's both good and bad, so the young animal shares youth factors with the old animal. And the old animal, it's been shown now several different ways. It helps them with their injury repair and muscle. It does all kinds of other great things for them. So young blood given to the old mouse is really great for them, and it improves their cognitive function as well. Unfortunately, it's two-way street here. The old factors going to young animal really impair them as well. So there's both great stuff in young blood and bad stuff in old blood. All right, and more recently, Saul Vieda's lab at UCSF did a really cool study where he used animals where they were actually the same age, but one set of the animals was exercised. And it turns out the exercised mice, if you take the blood from those mice and you give it to the sedentary mice, it helps the sedentary mice with their cognitive function. All right, so exercise is really great because even if you're an older mouse, it can actually produce a lot of these positive blood factors. All right, so a lot of you have probably heard something about the microbiome. The microbiome is one of these tricky areas because there's probably a lot of great stuff, but there's also a lot of hype about it that's not true. And so you have to really be a little bit careful sorting through the literature there. What is absolutely true is that we have bacteria in our gut, and it's a huge range of microorganisms, and actually changes all throughout our lives, when we're born, through middle adulthood, when we age. And part of the reason that it's important is because the bacteria themselves produce a lot of metabolites and process nutrients in positive ways when we have a good microbiome, right? And a lot of them, these microbes, the bacteria, can fight against invading pathogens like other bad bacteria and fungi. And it's also true that with age, our microbiome shifts, that we actually start to host more bad bacteria, and we also, we start to lack diversity. Instead of this huge range that we have in our young life, it becomes really like just a few microbes are in our gut. So it really is true that there is a very real aging microbiome, and it could be very poor for us. So there was a lot of hype about this for many years, and really only a few years ago did Dario Valenzano's lab do the right experiment. This is really great here. So what they showed was, this is using that turquoise killifish I told you about. So the killifish also have a microbiome, and you can see on these dots here, that's in the microbiome there. And what they did was they aged the animals to like a middle age. So that's what this 9.5 weeks is. And they either killed all the bacteria, that's this antibiotic treated, so the ABX, or they killed it and then added back young microbes from like a 6-week-old fish. So that's this here. That's the young microbiome, young MT transfer. Or they took it from like similarly aged. Okay, so that's the old. And so this is a great result. You see this green, that's the young microbiome transfer. Those fish live much longer. So this was really the first evidence that a young microbiome actually does something really great. And so I really applaud them for doing that experiment. And he went on to do other characterizations in this experiment as well. More recently, John Kreinen's lab did a really great experiment in mice. So similarly, they took young microbiome and gave it in mice. So this kind of gross, this fecal microbiota transplantation. So they took it from a young donor and gave it to an aged recipient. And what they really saw an improvement in the old mouse's memory and behavior. So I'm really excited about this result because it does suggest that there's something really positive about the microbiome. And if we can harness those differences, then we can actually really make a difference in how our cognitive function is maintained with age. So those are all very active areas of research. And so the reason I wanted to mention those is because, those alone are not going to necessarily come to the market. So what everyone listening to this is probably wondering, well, what can I do to live longer? And I would say we're not there yet, right? So there's lots of people giving you, diet advice and things like that. But what's really exciting to me is that these ideas from the academic lab is they're making it to biotech, which will then come to pharma and probably be available. Okay, so the first lesson here is you hear all the time about dietary restriction. I don't want you to leave this talk thinking that's the only thing that's important in longevity. For me, that's like one of 100 things, okay? One exciting area is that old drugs have been used for other things that are already FDA approved. They're already known to be safe. They're being tested in the aging field and then by these biotech companies to see if they can be repurposed for age-related disorders. And I don't have any personally, like any stock in any longevity biotechs, but I've been at meetings where I've been able to see some of the results. And it's really interesting that these are starting to thrive. And I want to emphasize, this is not just people talking about doing something or making money off of a podcast or selling a book or something. This is where they're really like doing things to put test drugs into clinical trials so that we can actually see if there's an effect or not. And I think that's critical for the field. So some of these drugs that are being tested in these clinical trials are things like senolytics and cenomorph. So these are drugs that target senescent cells. So those are cells that are dying and on their way out. And there's already been a phase 1 and phase 2 trials of senolytic drugs, and some positive and negative. And that's really important for us to know, like, do they make a difference? Those crazy blood factors I mentioned from blood plasma, those are being tested as well. And so that's a cool idea, right? You take a blood protein, you purify it, and then you give it to people so they don't actually have to take young blood. They actually just get that one factor that's really good for them. So that's an active area as well. You'd have to be living under a rock to not know that obesity and diabetes treatments are being not just tested, they're actually being used. And I would say, like, before I wrote this book, before I finished writing the book, it wasn't clear yet how wide-ranging they would be, but I would say these GLP-1 agonist treatments, like Ozempic and Wegovy and Mounjaro, those may have long-lasting effects on people's cardiovascular health in a good way. Just losing weight actually is really positive, and I'm really excited to see what the long-term effects will be, whether people will be able to keep the weight off and stay healthy, whether they start adopting healthier exercise or not. So, but I think that's already here. And so that's kind of interesting. Microbiome factors, we'll have to see where that field goes. And similarly, there's a lot of other drugs that are being piloted in these, in the research being done at biotechs to try to figure out whether any of them will have an effect on aging. Okay. And then of course, labs like mine are really interested in this idea of unbiased screening for new compounds, because we really want to see what could we do? And if we could test things, for example, on memory, would that be helpful, not just for old age, but for really important disorders like Alzheimer's disease? So it's actually a really exciting time to be in the field, because we're seeing the fruits of the labor that started in the 90s with identifying genetic pathways that can regulate aging, all the way up till now, where we can see that some of these things are being turned into really possible therapeutics that could help people. All right, so I'm going to wrap up now just with a few final thoughts. We're not done. We're still learning a lot about the regulation of longevity. A lot of times we're very surprised because we have these assumptions in the field, but not everything may be true. And so doing the hard work of the genetics in these model systems really helps us uncover those ideas. Again, I know it seems surprising, but what we find in the worm is often true in humans. I'm just going to tell you one story from my own lab. We've worked on memory in worms, and if you're curious about that, I can tell you how we test memory in a worm. But we found genetic pathways where we could turn on the pathway in an old worm. We convinced our friend Salveita to inject a hyperactivated version of that same protein into old mice, and it rescued their memory as well. So this conservation is really going to allow us to be able to find new ways to slow down aging and age-related diseases. And so that's going to help us find these drugs. Oh, I wanted to make this point. So again, back to the ethics question. Maybe we don't want to make, just like billionaires live to be 150. But one thing I'm very excited about is the idea that some of these longevity drugs could be multi-purpose. In fact, they could help people who are suffering from diseases earlier in life. So one of the most exciting stories I heard about recently was the idea of using a senolytic, so a drug that kills senescent cells, to help people who suffered from childhood cancers, who are now middle-aged and are suffering from a lot of early aging effects. So that's exciting, right? So that's not, doing something weird to help people live absurdly long. It's just helping people deal with a lot of age-related diseases that they otherwise wouldn't have had. Okay, if you learned nothing from my talk, I would encourage you to exercise, because there's almost nothing bad about exercise. And a lot of the pathways that we find to extend lifespan actually are mimicked by various forms of exercise. And finally, I'd like us all to remember that, at least I hope the field won't all go in the direction of helping only the super rich people. Like a lot of the things that we could do to help people live longer, we really need to make sure there's some equity in health treatments. So that we can not just raise like the maximum lifespan, but really help increase the median lifespan of everyone. And my last point is, I'm not a climate scientist, but it's not hard to see that if we don't do anything to invest in the planet, the world we live in now, there's going to be really no point in living long. Right? So if you could live to be 200 years, but there's no great planet to live on, then there's kind of no point, in my opinion. Okay, so with that, I'm going to finish, and I want to thank you all for listening. Probably maybe a little too much science too fast. I apologize for that. I get excited. But also I'd be happy to take any questions you might have.
That's really fantastic stuff, Coleen. I'm sure everyone's been very excitedly listening along. And I mean, personally, as a scientist myself, I love that the book was really unabashedly sort of science driven, and it's wonderful when you see these moments of where the scientific community really comes together over big discoveries, right? You mentioned the one about DAP2, doubling the life of an organism is super cool, right? You mentioned in the book, induce pluripotent stem cells, and turning cells back in time. That's super cool, and everyone comes together on these sorts of things, and I think that's really cool. And one of the things that you really harped on in the beginning of your sort of introduction is that you're unapologetically in support of model organism research. And I wonder if I could, while people are writing some questions into the chat here, if I could ask you, to expand on that a little bit. When people say, "Oh, why do you bother with flies and worms?" What do you say back?
Well, for certainly, I mean, there's no place, maybe developmental biology is another place, but really the aging field, I think, would not have gotten where it is today without the existence of research in worms and flies. It just wouldn't. Um, I mean, lots of the problem is when you read a newspaper article about something in mice, they always make it sound like it was discovered first in mice, or worse yet, in humans, right? But the truth is, a lot of this work was done first in these simple model systems because they're so easy to work with. They're cheap. There's no ethical issues, at least I hope not, with, we use like 1 million worms in an experiment sometimes, So it just, we have learned so much that we would never have been able to do in an organism that lives 2 years, right? It's just impossible. And so it's really accelerated our rate of research, and we know a ton of, and so, yeah, I harp on it a lot because even other scientists, I was a postdoc, so it was probably the early 2000s, and I went to an aging meeting, and there was a panel discussion, and everyone on the panel, there was an epidemiologist, a gerontologist, an evolutionary biologist, and they all kind of made fun of model system research. And I actually, as a postdoc, had to stand up and say, look, you're working on an insulin pathway. You did a study that discovered like an insulin receptor in centenarians. Like, all of them benefited from the work that we had done in C. elegans, and they wouldn't have done it. And so then they called me model organism. I was like, whatever. It's like my super.
Yeah, you need a T-shirt made now.
Exactly, a cape. But it's fine because like we need to do it. But it is true. I want to explain this also, especially to U.S. audiences, because a lot of our research is funded by the National Institutes of Health, which is funded by taxpayers. So they should know where their money is going, and they should know that it's being used responsibly to try to find ways to help improve our health. It's not just dumped down the drain. And working on a worm or fly doesn't mean you're working on something just esoteric or just for the heck of it. We're actually finding new interesting stuff.
Yeah, absolutely. And so I guess on the other side of that, you mentioned the sort of the way you've got all these cool clinical trials happening in aging drugs and this sort of space, but that's got to be really hard to monitor, right? Like outcomes in a clinical trial of old people, right? Like, these are going to take decades to really pan out. Is this really good for us? So how does the field deal with that?
Actually, this is where, like an academic scientist, I don't have a biotech, I don't have any, but I think this is where it's interesting what biotech companies are doing, right? Because they don't have the time and money also to look at how aging works. So what they do are find really important age-related diseases and proxies where they can monitor outcomes, okay? And so this is important to do. You can't, like, the problem for me, I'm just gonna back up and say, you can buy 1 million nutritional supplements online and stores, and there's no evidence one way or another what's in them or whether they work. And so the only way we can get to a real answer about that is to treat them like a medicine. You have to be tested. You have to go through clinical trials. You have to report whether things were safe before you can go on to the next level of reporting whether they're efficacious. And so the only way this will happen in the field is if these biotech companies actually go to the effort of doing these trials. And so they're doing that now. And I really applaud them for that, because somebody has to do it. I mean, somebody should do it. Other people are just selling supplements and making a lot of money off it irresponsibly. And so it's really important that people know, and that's why I want them to read the book and understand the research, because I want people to make smart choices about what they spend money on and what they put in their bodies. And these supplements, they might not, like, what's in the bottle may not even be what's on the label.
Yeah, no, absolutely. There's a lot of quackery out there.
Yeah.
I'll move to a question from Alan, and Alan asks, what are some of the opportunities maybe that artificial intelligence, modeling, machine learning might offer for the study of longevity?
I think that one of the most convincing areas is in drug design. So artificial intelligence is very useful for things where you can have like a good model data set to start from. And so this is where compounds are useful. And that's actually a very active area of research and some companies are doing that as well. So I think AI is going to really play an important role, because it's really hard to screen enough chemicals to know what you, but if you could do all the in silico screening first before you narrow it down to a few things that you actually do experiments on, that's a really good use of AI, and that's actually going on currently.
Yeah, no, absolutely. And then I guess something that people might be wondering in the crowd, right? You know, we probably don't have a bunch of 18-year-olds watching. You know, how much of the damage is already done, right? You know, when you start doing these things to turn back the clock, you know, you sort of caloric restriction, reduce your sugar intake, these sorts of things. Are we already doomed at the age of 42?
I love it. Such a you know, non-American question. Americans are like so
There's
optimistic.
always a drug.
Let's see. Yeah, so look, this is where some of the responsibility in how we report our work also comes up. Like, I do think that we have to be responsible in telling people what can and can't be done. And so the word reversibility gets thrown around sometimes when it shouldn't. What I will say is right. So there's damage that we've done being out in the sun, right, for a long time. How much can we, but there's always a possibility of slowing damage, like further damage. And in some cases, like, as you know very well, like if you could take a cell and replace it, you might actually be able to reverse it. So it kind of depends on the system you're talking about, right? You know, actually, I had a very optimistic postdoc. We're about to publish this. It's like, you know, you don't often publish negative research, but we're going to in this case. He had the very optimistic viewpoint that maybe we could reverse cognitive aging in C. elegans by turning on the Yamanaka factors. So these transcription factors to like basically reprogram old neurons. But, you know, it didn't work. It actually gave the worms had tumors in their head, and it made them dumber. And so I think it's important for us to sometimes put out some of that research so we have like an idea of what would be the consequences of doing this badly, instead of just always saying, oh, just do anything. And so we have to do these experiments. So there may be things that we can reverse. Let me just say, there may be things where we can stop the aging and slow down aging. A very few cases may be reversible. I think we just have to, you know, do more of the research to figure that out.
Yeah, and it touched on a point that's near and dear to all scientists' heart, right? Like it's not easy, right? And if it was easy, it'd be done.
Yeah.
And a lot of this stuff that you test fails, right? And it's a
Yeah.
real journey to get to these big discoveries that, you know, will improve lifespan or reduce cardiovascular disease. And so yeah, I think that's really tough. You also mentioned near the end of the book this sort of kind of debate about immortalists versus realists, and inside of the scientific community, these, you know, the sort of hype that goes along with, you know, we can live forever, and what you read on the news, you've mentioned a few times, like, you know, you understand that people are reading these articles. So wondering, you know, where does that all go? How do we improve responsible journalism? How do we make sure that people can get, you know, trustable evidence? Where do they go for that?
Yeah, it's a little frustrating as a scientist is actually working in the lab, because what gets into the news is often not the work that we do, because that's considered a little boring, right? It's the hype people, and some of those are not even scientists, right? They're just people who like to talk about aging, and so they're considered experts just because they talk a lot about aging, but they actually don't do any research themselves, and they make outlandish claims and promises to the public. And so if you as a scientist say, hey, I don't think it's responsible to say that the person who's going to live 500 years has already been born, then that person who's saying that stuff calls you like you're against longevity research. It's crazy. And so, you know, it's frustrating in the aging field that being responsible sometimes means that you have to say, well, there's limits to what we might be able to do, at least based on what we know now.
Yep. Yeah,
But you
absolutely.
know, we're not always the ones that journalists want to listen to.
Absolutely. Well, Coleen, it's been absolutely fascinating speaking with you, and I hope everyone else has enjoyed the conversation as well. I want to remind people that if you did miss out on any chunks of this, there will be a recording of the event, and that will go for other people who haven't tuned in today as well. This will be available on the Festival of York's Ideas or Festival of York Ideas YouTube channel, which can be accessed from the Watch Again section of the festival. on the website. And you'll also be contacted by email when this video is available to view. And then finally, as we thank Coleen, also of course she's not here without some way of repaying her, which is that if you do want to purchase a copy of Coleen's book, How We Age: The Science and Longevity, it's available from our partner bookseller, Fox Lane Books. And I can say from personal experience, it's a really fascinating read. Although you do need to be prepared for the acronyms, because we can't avoid those in science. But Coleen really brings it down to the level where you can actually engage with it as somebody that's a non-specialist. So really well done to you, Coleen, and thanks again for joining us today. We really appreciate it.